It is my great pleasure to introduce tonight's speaker, Dr. Manuela Dal Forno. She was a resident of our area a few years back and is right now at the Botanical Research Institute of Texas. Dr. Manuela Dal Forno is a Brazilian-American researcher interested in multiple aspects of the lichen symbiosis. She earned her PhD in environmental sciences and public policy at George Mason University in Virginia, and later was awarded 2 postdoctoral fellowships to work at the botany department at the Smithsonian National Museum of Natural History. She joined the Botanical Research Institution, or BRIT, in 2019, where she is currently a research botanist and the Lichen Evolution and Diversity Program leader. Her research has particularly focused on the discovery of new species, especially in the tropics, and how they are related to one another. Please join me in welcoming Dr. Manuela Dal Forno.
Thank you, everyone. So I'm going to start by sharing my screen. So just bear with me 1 second. Let's see, share. Okay. Okay. And you can see, okay, presentation mode.
Looks good.
Yes.
Okay, let's get started. Okay, so thank you, Serenella, for the introduction and for the invitation to be here. I am Manuela Dal Forno, and today I'm going to be talking about my research on lichens. And I just want to start by defining who I am. In the introduction, I think you had a very good idea already. So my lichen lifestyle is defined by 4 different main activities that I perform in the field, in the lab, also doing outreach and in the office. But regardless of where I am, I consider myself a lichenologist. So my talk will be not divided, but will have a flow that I will start with a background in lichens. I will present the bulk of my research in integrative systematics, and then talk about some of the projects that I did when I was a postdoc at the Smithsonian, talk a lot about the collections and how much data we can get from these collections already deposited at museums worldwide. Talk a little bit about my new love, which is lichens from cemeteries and monuments. And then also just end with a little bit of some common lichens in the DMV area. Starting then with defining what are lichens. They are, as the name says, symbiosis literally means living together. Lichens are complex units of, symbiotic units, and they're formed by a main fungal partner or the microbiont, also a photosynthesizing partner, the photobiont, here represented by a green algae, but about 10% of all lichens could also have the photobiont, a cyanobacteria, as well as a diversity of microorganisms that form the microbiome. So what we see in nature is the result of these. So it's what we call the lichen body. It's the proper name for that is the thallus, which is like a mini ecosystem. So now if you go inside of the lichen thallus, you see that in this really beautiful image here, you see that the hyphae, or in this very light beige color here, you see that the hyphae of the fungi is wrapping around the cells of the photobionts, and they are penetrating it with haustoria. And you may at first think, oh, lichens may, just optimally live in a beautiful, pristine tropical forest, or, you just come back from a hike and you see beautiful rocks covered in lichens, but they are present in all terrestrial ecosystems on Earth, and even on some aquatic ones, like especially on freshwater, but even in coastal vegetation, maritime ecosystems, and extreme environments such as Antarctica. But good for us, they're also right here in the city. And I usually hear many people, mention or make a comment when I say, "Oh, I study lichens," and they say, "Oh, those small little things growing on the tree." And I say, "Well, some lichens are small, but others not at all." And then you can see here, I am at the scale of this road bank in Brazil, that all of this whitish stuff that you see, these are all lichens of this species called a reticulifera. And you can see that this is a very important species of lichen, and it's part of the vegetation of these, and is definitely helping to hold the soil together here. So it's very important against erosion for this environment. And very similarly, here we see these gorgeous cladonia mats growing in a scrub, and this one here, not here, but in closer in Alabama, and is helping to maintain the soil structure of this very delicate sand ecosystem, also that in this case is reducing the erosion by sand. And in other environments where these cladonia mats are very predominant, such as in this taiga, or it could also be in tundra, lichens are very important for the environment, and they serve as food for animals such as reindeer. But in for other environments, such as some forests in Yunnan, in China, for example, where these snub-nosed monkeys that you can see eating these beard lichens, they spend most of their lives living up in the canopy, and they have adapted their lifestyle to just live there because they have access to these lichens to eat there. So that is one of these extreme environments, because they can do that. So this is a case where they have evolved because they're able to get most of their food living there up in the canopy. So lichens have been part of the evolution for these species. So this is really exciting. And there's a million cute pictures of these monkeys online eating lichens. So some of them even like feeding the baby ones. So you have to see these pictures. I show you, it will make your day much better. And of course, aside from that, so aside from a lot of different animals that eat lichens directly, there are also a lot of other species that utilize lichens for nest building. And there's over 50 species of birds here in North America that utilize, as an example, here you see the hummingbird, but also a lot of other animals that use lichens as mimicry or for camouflage. But besides the main components that I showed, of the lichen that have over millions of years of evolution, including the microbiome, that have helped lichens to adapt to such a diverse, diverse ecosystems. The chemistry is also very important for lichens. That's probably one of the first things that when we read about lichens, we learn So this yellow pigment that you see in these images here, they're called anthraquinones, and they're present throughout an entire family, Teloschistaceae, and they have photoprotective properties. And basically what they do is that they're acting like a sunscreen to the lichens. So they're able to live in environments that are very open, protecting then the photobiont layer. And in other examples, you can see here this beautiful red lichen here, Marcellarium purpurina. This lichen is growing on exposed tree bark in tropical lowland forest. And similarly, these pigments are being produced only in these parts that are producing the spores. So it's also protecting these reproductive structures which are going to have the spores. So it's also have an evolutionary function. And the other two images that you see there on top, Graphis dendrogramma, is an example of a lichen that grows on a dry tropical forest and is producing hydrophilic substances. And if you see, I hope it comes through that this lichen is slightly yellowish, and it because it has this acetic acid that is producing a and it's a hydrophilic substance, while at the bottom, stegobolus is producing a substance that is hydrophobic. And in the other hand, so on top, you see a substance that is being produced that is hydrophilic, and that lives on a dry forest, and at the bottom it's in a wet forest and is hydrophobic. So it's really amazing to see how these chemicals are there also helping these lichens to survive to these habitats. And while some of these chemicals, we have an idea why they're being produced, as an example of this adaptation to dry and wet forest or tropical forest, some of them we have no idea. So here, going back, this is the same lichen in the normal light, and this is the UV positive. So what we do is just expose this lichen to fluorescence. It's just in one of these UV lamps, and you can see that it has this UV plus reaction, and that represents that it has lichen xanthone. And why is this lichen UV plus yellow? We have no idea. So lichen research has advanced a lot, but we don't know everything yet. So there's still a lot of work to be done. So you've seen this image before about this Cladonia mats, but did you notice that it had two different colors for the species? Or you just saw one big thing before. But now if you notice and you pay more attention, you see that there's two different species here. And again, I hope that the colors, you're able to see that they're slightly different, that there's one that is more grayish and there's one that is more green yellowish. And that is already indicating two different species just because of the colors. And they indicate that the grayish one has a tranurine that is a different chemical profile than this yellowish species that has oxalic acid. So just by looking at these previous images in all these beautiful pigments and acids, you can have an idea of how diverse lichens are. And the current numbers is that we have is that there are about 20,000 known species distributed in about 1,000 genera and 115 families. But to add to this very complex world of lichens, they do not all come from the same common ancestor. And I love talking to y'all about lichens and these because I don't need to explain to you that there's Ascomycota, Basidiomycota, and a lot of other fungal groups, but there are lichens in both Ascomycetes, about 99%, and only 1% in Basidiomycetes. So this lichenization, as I started talking about from the beginning, it's a fungal lifestyle that has evolved multiple times throughout the fungal tree of life. And when we're talking about total number of species, given that I think the most recent number that I got is that about 155,000 species of fungi are known. So that this represents about 13% of all fungi. But despite the many advances of species discovery overall, the estimated is still much, much higher. So there's a huge knowledge gap in fungi. Of course, we know that we're not the only group that is still very behind if you think about insects, for example, as well. But we're still very behind. And many of the missing fungal species are suspected to be only in these cryptic taxa, detectable only through sequencing techniques. But we know that even presumably well collected taxa are also substantially, there's still substantially undescribed diversity. Even this, big showy lichens such as this lobaria that we collected just a couple years ago, that might even be a new genus. So it sounds like a lot of work ahead of us, not only for lichens, but for all of the fungi. So our main challenge as a community, though, is about the global biodiversity crisis, that it does stand as one of the most pressing and immediate threats, especially in the tropics, because of this a lot of, this laundry list of terrible destruction that we see on a daily basis, especially in the tropics. So in addition to these, the habitat destruction, the climate change, invasive species, and these threats that we see every day, the problems are especially acute for groups that are less studied, such as lichens. And they are, while lichens are ubiquitous, they are relatively less studied compared to others. So it is very important that we have a broader understanding of where some of these unknown diversity could still be discovered. So this way we can potentially target some of these areas before they go extinct. So this is some of our goals, why we started to combine study of herbaria with boots on the ground, how it's known, just to go out there, collect as many areas as you can. So long story short, there's a lot of lichens out there, but just not a lot of lichenologists to get all of the work done. So as much as I would love to go anywhere in the world to keep collecting, I still need to try to use some of these lichens that are already in herbaria and just try to target areas where I still think that a lot of these unknown diversity could still be found. So this way we can prioritize the time and the funds that we have to identify some of this unknown diversity. So this is the base of the research that I do, is collections based research. So just to really find the basis of all we do is to describe species. And why is this species important? We were just going through, because we eat certain things, because we want to put names on things, because it is when we are studying about everything that I do, systematics, interactions about microbiomes and photobiomes, the microbiome, metagenomics, everything that I'll show you here. I want to know, if I'm comparing something, I want to make sure that I'm comparing apples to apples, not apples to oranges. So knowing what is a species is very important for me. So it's the basis of everything that I do. So given the importance of the collections, I really wanted to give you some idea about collections. I know that the focus of the group here, it's more practical, it's learning to be out there in the field. But for me, the work that I do is also very important that we have vouchers for posterity and the reproducibility of my work. So when I go to one of these trips and the field work that I will also show you later on, I always have collecting permits so I can legally collect and maintain this material. And we keep them in these envelopes and deposit them in herbaria. So collections, lichens have been collected as fungi for decades, and they have been housed in herbaria and Bulgaria in worldwide. Historically, they have supported the study of morphology, species delimitation, geographical distribution, and all of these studies about how plastic the species can be regarding their phenotypes. But now with molecular studies, some of them have challenges our previous concept, but they have also been able to offer new insights. So now we can test a lot of new hypotheses combining both of these data. And where are the biggest herbaria in the world? Long story short, in Europe, because of historical, long time ago, Europeans, they already had a lot of money to go all over the world and deposit the specimens and so forth. You know all of these. But here are some very interesting numbers about the lichen herbaria in the world and how some of these numbers are really, really high. And where are we in the US about this? Not too far behind. The largest lichen herbaria in the US is New York Botanic Garden, followed by Harvard, so the Farlow Herbarium, and then the US, which is the closest one to you. And then after that, there's a couple other ones that are important, such as the Duke Herbarium and Field Museum, for example. I put the specimen there. That is my favorite specimen from the US. So since we're talking about collections of lichens, I believe that to enhance and to connect the past and the future, I really do believe in not only making new collections, but also adding value to the existing collections. Is the real, my earbud just fell, excuse me. Is the way to move forward. So in all of the research that I do, I practice what I call integrative taxonomy or integrative systematics, which simply said, it combines multiple lines of evidence. So morphological characters, molecular characters, ecological, and computational. So to try to comprehensively study the evolutionary relationships and try to classify all of the lichens that I study. And here are some examples of the type of data that I take into consideration when I am talking about species. And the group that I have been working on the most for the past 15 years is the basidiolichens, the 1% of the total lichens. And here we can see we have a lot of different images. The first one is Multiclavula musida. The second image here is Susbaheromesis. Third one is Lepidostroma, and then we have Semiomphalina, and then all of the other 5 are the subtribe Dictionematini, which is the one that I'm going to focus on. The Dictionematini is a group that is found worldwide, but it has its highest diversity in the tropics especially in montane forests. And I hope that you can have an idea of the habitat that they prefer just by looking at these images. So they can be either in the neotropics, the paleotropics, and just by looking at the color previously, you can see, I can go back, that they're sort of bluish. So they're not only basidiolichens, which is 1% of all lichens, they're also cyanolichens, which means that they have a cyanobacteria as their photobionts. And these lichens have been largely known in the community. They've been, wildly collected, especially in the neotropics. And then there were many names available. Some of you might know Parmasto. It was very known in the mycological community. And in an attempt to monograph of, many names available and genera, he worked with over 1,000 specimens from all over the world, but Parmasto was a mycologist, so he used entirely mycological characters, and he decided to synonymize everything that was known for the group into a single genus and no more than 5 species. So as taxonomists, we love putting names on things, and with Parmasto's system, it became very easy to identify. However, with so few names available, it became very easy, but it also became what we call a taxonomic trash can. With all foliose forms, would only go under one name, Dictyonema glabratum or Cora pavonia. No matter if you collected in Terra del Fuego or Florida, it would only go to this one name. And the same happened to filamentous forms, especially here in the neotropics, there was only one name available. However, we were out there and we were looking at these differences in the field. And could this be indicative of different species? Look at this, all of these beautiful images that you're looking in your screen. All of these were considered one genus and just 5 species. How could this be possible, right? That was our question. So this is how I started my PhD. How diverse is really this group? Because we imagined that there was no way that was it. And this is when we started, making all of these phylogenies, inferring relationships, and we found out there were actually 5 genera and hundreds of species. And I'm not gonna go over these because this now we have a lot of things that are out there, but we wanted to go further. We've been able to advance a lot from 5 species. We were able to resurrect a lot of the 40 of the species that had been previously published, all of the genera too, we were able to resurrect. So now we have about 160 species published, but we have a lot more detected and even more estimated. And we also have the distributions and the morphology very tightly knit with their phylogenies. But we still see a lot of huge gap, again, between the estimated species and the detected. And we started to think, then how can we fill this gap with herbarium samples? And this is how I started my postdoc at the Smithsonian. They had one of the largest collections of this group that I work with over 400 samples of spanning over 100 years. And I decided to test a couple different methods, which I'm going to go over in a couple more slides. But one thing that, since now you have the Lichen lab, I thought it would be funny, or fun, not funny, to go over these. So a lot of people ask, right, how do you work with DNA with lichens? Because there's so many things. So you start with a piece of lichen, you extract the DNA, and then you select for what you want. But there are a couple other techniques that you can do that you can select for multiple things at the same time. One of them being barcoding, that you can sequence all of the fungi present in your sample or all of the bacteria or all of the algae, but you can also do metagenomics that in one sequencing, you can sequence everything, or you can also try some culturing. But today I'm going to be focusing on high throughput sequencing, or also known as next generation sequencing. And there are multiple ways to explain next generation sequencing, but I will explain in a way that I find it to be the most simple way. There are two separated types of high throughput sequencing. One is called amplicon sequencing, in which, if you follow me in my image, you will select one piece of the DNA that you're interested. Okay, so you have a lot of DNA in your sample. So in the amplicon sequencing, one example is the metabarcoding. So the amplicon, you're selecting one piece, and that one piece you're gonna extract for all of the different types of DNA that you have in your sample. So, but it's just that one piece. And then for the shotgun sequencing, or for example, the metagenomics, you're not selecting any piece. It's shotgun sequencing. You're shooting everywhere, and then you're gonna get what you get. Okay, so that's an example now of the metabarcoding. You have a sample of the lichen, and a lichen, it's like an environmental DNA. You're not culturing anything, you're not isolating anything. So you start with a lichen, you're extracting the DNA, and when you have the metabarcoding, you're gonna get a lot of different DNAs. So you're gonna get the DNA of the lichen, but you can also get pieces of DNA of maybe if the lichen is growing on the soil, you may also get some sequences of soil fungi. You can also get some sequences of a spore that is living nearby, and the spore landed on the piece of the lichen. So with metabarcoding, these methodologies are very sensitive. And even if there's one spore, and you extract that piece of lichen, sometimes you wash the lichen, but sometimes they're still gonna get there. So with metabarcoding, you are still going to, if you're selecting for ITS, which is the fungal barcoding, you're still gonna get a sequence for all of the fungi present in your sample. So with that, hopefully your sample is still going to have the majority of your sequences belonging to the main microbiome, which is what happened to the lichens of the museum. That out of the 418 sample, I was able to get a positive microbiome sample for 75% of the samples. So the other 25 were either contaminations, or either I couldn't amplify because the sample was too degraded, or for whichever reason. But for 75%, I was able to get a sequence for the lichen that I was interested in. And then I selected only the sample for the microbiome, and then I infer the relationships of those samples to everything else that I already had previously acquired samples. And you can see these tiny little tips here in blue are just the samples from the museum. So when I put in a bigger framework, I was able to compare the samples to everything else that I had. And with that, I detected that by adding samples from the museum, I added samples with sequences from 11 countries that I didn't have data before. I also increased the number of specimens I had per country. For example, Panama, I only knew actually, I only knew one species from Panama before, and I now knew 6. So I don't know if Panama still has those 6 species of Cora, but I know that at some point it had. I also was able to expand the geographical distribution of some species. I don't know if you can see on the corner of the slide, there's a picture of Cora itabaiana. I only knew that species from one location in Brazil. But by adding herbarium samples, I was able to see that this species not only occur in Northeast Brazil, but also in Suriname, Guyana, and Trinidad Tobago. And what all of the Cora specimens that were all under just that one name before that I explained at the beginning now belong to 75 different species in 2 genera. So all of the samples just had one name before in the herbarium, and now they have 75 species. So that also made the herbarium a lot more important for this group. And the herbarium now has a lot more species than it previously had. So imagine that for each group of lichen or plant or whichever group you're working with, if you start digging in, there's still a lot of treasures for each one of these samples that we work with. And a lot of new species. So 25 new species are just discovered in the herbarium that we don't know if they still exist in the wild. And this is an example of what is the work that we had to do. So all of these were just under one name here, Dictyonema glabratum or Cora paphonia. Now we have to go and rename each one of these samples. And one of these now is a new species, Cora timucua. And we were able to sequence this species and find out that is a new species known exclusively from herbarium sample. And now we have over 30 specimens, and this species only occurs in Florida, but we haven't seen or heard about this specimen since 1985. And this was a big deal. It even was part of the news. You know, it was in the Washington Post and a couple other media, because it's a huge deal to discover a new species and you find out that it might be already potentially extinct. So we named it after the Timucua people that used to also live in Florida. So that with that, we were also able to work with IUCN and describe it as critically endangered. And excuse me, let me just have a sip of water. And we also have a iNaturalist group. So if you're ever traveling to Florida, you have to look for this. And if you find it, take a picture and tag as potentially Cora timucua. So anything, if anybody tags Cora from Florida, it will pop up into this project, and then we can actually check if it's this species or not. But don't collect it because it might be the only one. But we do have a hope. We're applying for funds, and we actually might have a lead with an NGO that might fund potentially a community project to try to relocate these species in the wild. So there's still hope that we will find these species. And so combining all of this information, we can now try to find what are the areas that we don't have any data and where we should focus our efforts. So just to finalize this part, for example, I don't have any specimens from Nicaragua, Paraguay, a lot of places in the Caribbean, Belize, and El Salvador, I think. So anyway, oh no, El Salvador, yes, I think I was able to find a few. So anyways, this is the way that we can combine all of the information and find maybe our next places where we should go to try to find for these areas. So now jumping into another method, the metagenomics, I will explain this by utilizing an analogy of a puzzle. And your sample in nature, think about it's like a whole finalized puzzle, right? So you start like this. When you perform the shotgun sequencing, it's like you shear the DNA in a million pieces. So imagine like a very, very big puzzle. However, since the lichen is a multi-species organism, it's like having a a box, not having one puzzle in your box is like having 12 or 500 puzzles in one box. But you need to not only figure out, but they all come in just one bag. So you not only need to figure out to, where to each piece where it goes, but to which puzzle it belongs to. And there are different ways to do that, right? So you can try to find each piece to goes with where, or you just try to find you know, does it go here? Does it go there? So there are many ways to do that. And the same way with the bioinformatic analysis. So here's someone trying to attempt to put them into different bags based on the image, based on the color. Some people start with the corners. So there's many different ways to try to do these pipelines. And that's why there's so many programs. And eventually, sometimes you will have some missing pieces. And then there's also ways to deal with that. So working with metagenomics of lichens is a very similar. And one of the programs that I use is this Busco, which I'm not going to go into details, but the way that this program or software goes is that it evaluates the presence of conserved single copy orthologous genes across different lineages. So in this case, I'm going to show you the percentage of Busco fungal genes. What does that mean? It means that there is a data set online available that people have created that have located that 758 genes are present in all fungi. So if it's a fungi, it will have all of these genes. If it's bacteria, it will have another set of genes. If it's eukaryote from another group, mammals or these. So you can create these categories for different lineages. So for the one here, I selected to show you the fungi. And then the Busco categories. So you have this puzzle, right? So each one of these lichens was sheared into many pieces, try to put them together to say, this is the microbiome, this is the cyanobacteria, this is the other bacteria in the microbiome that are part of. How much can we assemble out of these genomes to create this Busco? And that basically means how much of the genome can I put together based on these samples? And I wanted to compare not only fresh ones, but also samples from the herbarium. So I just wanted to show you that we were able to get a lot of high numbers with these as well, including from herbarium samples. So again, not going to go much into detail. I just wanted to show you a lot of the potential from herbarium samples and also everything that can be done with lichens. And this is all being done with fungi as well. And the final one that I wanted to show you is the fluorescent in situ hybridization, short for FISH. And this way, you can not only know the presence of bacteria, but also the location of where it is. So going back, so this is Dictyonema, and if you look really up close, all you see is this tiny little filaments. So you can imagine like the hyphae of the fungi, or in this case, the hyphae of the lichens, they're wrapping around the cells of the unicellular but chain forming cyanobacteria. And the way FISH works, that there is a probe that will hybridize to the material, and there are different dyes that will bind to the DNA. So you can say, oh, if it's a bacteria, it will bind it to the bacteria and will produce a dye that is green. If it's this other bacteria, it will produce a dye that is another color. Just so you not only see, because with sequencing, you can see, oh, all of these bacteria are there in the lichen. but you don't see where in the lichen they are. You just see that they're there. But with this technique, you can see where they are. For example, here, you can see all of these bright things that you see in red. You can see there are bacteria that are there. And then, so these are some comparisons of specimens that in 2016, they were fresh at the time. And then at the bottom, you can see that it's a sample from 1981. So you can also use herbarium samples. And in this case, I am showing the photobiont in green. And then the orange here, there are some alpha proteobacteria, and then there were others, there's some other type of bacteria. So I just wanna show you that we can also try to see how much of the bacteria have remained over time in these lichens. So just to finalize this part, just for each sample, then we try to combine all of this data from the field, the morphology, anatomy to create a working name, which is not different than a hypothesis. We add the phylogenetic data, and then we bring everything together to create uh, um, a concept about that species. So, but there's no one size fits all for describing lichen species. But to further explain a little bit about the work, we just finalized a grant working in the Philippines, and I know I'm already running a little bit short on time, so I just want to quickly go through this. Is a project that we have a lot of different goals, but basically we wanted to find out, my component of this is to find out how diverse are the lichens of the southern Philippines, documenting the lichens while there, bringing everything legally here. We have all of the lichens here at Brit, and then we also made copies that are kept in the freezer for the DNA. And then you'll see a video that is made by one of my students about the latest expedition. We tested a little bit before the meeting, and there is no sound coming through, so I'm just gonna explain it over. It's a little slow, but we thought that you still have an idea of what's going on, so. So for this project, we collected everything. We curated all of the samples at night. You can see sometimes is very dark at night, but we would go to our tents at night only when everything was, curated, typed in. I brought my computer to the field every night, but when samples arrived here at Bridge, all we had to do was to print the labels, curate, but everything now is digitized and is going to the Lichen portal. So we had a total of 6 expeditions of about 1 month in each one. And I will share with you now some of the final numbers. We collected over 13,000 samples. A lot of these, most of these will be able to have duplicates. As I said, we collected everything, not only the big, showy, pretty things, a lot of crusts. Gladly, a lot of us like crustals, lichens. We fill out our tents, we hang things, we also pressed, especially folliculus lichen crusts. They look so much better when you press them. And we were able to train a lot of people, visit a lot of communities. We always had ceremonies with the local people, and we also had closing events with them. Everybody had to share something that they learned, and it was a very enriching experience. And we're writing another grant, sort of like a follow-up. And meanwhile, we can reflect of how our work with lichens have touched so many lives, and sometimes in very unexpected ways, because after we left, a week later, we received the news that one of the local researchers that was with us, he named his new baby lichens. So that's how much he was impressed that he lived by that forest, his whole life. And he passed by those woods every day and he never knew what a lichen was. And now he will never forget what they are. So he named his baby lichen. So, and also very important in these expeditions is training the next generation how to properly collect, and also how is important to also leave the specimens in the countries that we visit. With that, then I'll switch to very quickly our collaboration with the U.S. National Park Services, which has awakened inside me another passion for lichens in monuments and cemeteries. You might have seen this, that there was a couple years ago, there was wave of cleaning of the sea monuments. So I collaborated with Judy Jacob, from the U.S. National Park Services, because they were starting to clean and we wanted to have inventories of what lichens were there. And together also with Helga and other volunteers at the Patuxent Research Refuge, which is part of the U.S. Fish and Wildlife Service. I was also volunteering at in this park, and they had a lichen project. So they had some of the oldest cemeteries in the area. So as you can see here, and we started looking at the graves there, and I found out that is, that besides lichens, they also cemeteries and monuments also accumulate a lot of culture and history. So I started visiting, and it's really amazing how you can have full lichens there, and this is something that you might find in the area. So Xandoparmelia conspersa, it might be one of the most common lichens that you see. So just as a brief reminder that we have three main types of lichens. The crusty, as the name says, they're very crustals, very hard to separate. So when you collect, let me hold it like this, very hard. You have to bring the substrate with you. So in this case, it's a pink marble that Tom might recognize. It's from Maine. So you have to bring the piece of the substrate with you. Leafy, it's very leafy. I think, I'm sorry, I cannot see it now. Who said it? The kale of the forest or something like that before. I often think that these, leafy lichens, they look pretty similar, leafy lettuce. So they are the foliose types. And we also have the branching, the fruticose types, that it's usually pretty easy to remove, as long as, of course, you have a permit and you collect with the attachment. Okay, so if you're collecting a fruticose lichen, you need to collect with the attachment because it also contains very important information. So the branching fruticose type is sort of your 3D lichen. So the fruticose lichens, they are pretty common, except they're not common in the city. But if you start going further out in the city, you will start seeing them more. The three main groups that you see there are the tape lichens, the Ramalina. They're usually flatter. The beard lichens, Usnea, I think I saw it one time in the DMV area. Same with Ramalina. Oh, I'm lying. I saw some Usnea and Ramalina in Tacoma Park in last year. So there are some around. And also Cladonia, I've also seen some around, especially in parks, a little bit further away from the city. But maybe I'm sure that some of you already might have a list where to see what in DC, right? Maybe we can exchange some ideas. But this, for example, in Patuxent, you can see this whole Cladonia patch here in some of these cemeteries. That's why one of the reasons why you gotta love them, because there's absolutely beautiful. So this is the type of reindeer lichen. So Cladonia, they have many different common names just based on the shapes. The Parmeliaceae, I think these are the top four most common Parmeliaceae that you see there. The Flavoparmelia caperata, you see it's sort of your wrinkly gray, yellow green type on the trees. Um, and then you also have Punctelia rudacta. Even though it's more sensitive to air pollution, these two are from the mall area, so very close to the monument, the Smithsonian museums. Both of these pictures are from downtown DC. And then you also have the Parmotrema hippopotrum that we were just hippotropon, excuse me. We were just talking potentially someone collected there. It's one of the most common species there. It's very fluffy, it's very ruffly, and it has the white borders. And then the Parmelia sulcata, again, very common, and it has sort of these lines, these whitish lines that you see here, while the Punctelia rudacta is also more bluish and it has white dots. So these are the most common big foliose lichens that you see here. The something else that you might see, not so much in the city just because of the rocks. So unless you go to a place that is rocky, the green shield lichens, very similar to Flavoparmelia caperata, is baltimorensis. The only difference is that it's growing on rock. So I just wrote it down here, very important, substrate where it grows. So when you're taking a picture, maybe taking a picture further out about where is it growing is also very important, or making a note, something like that. The special characters, we just saw the Punctelia rudacta. This is what I meant about the small dots here. These are two other species that I took pictures from the DMV area, Pixini subcinerea, Pixini soreggiata. They're pretty similar, and they both have the same common name, mustard lichen, because the medulla is sort of mustard in color. They're pretty similar, so if you have one or the other, I would say that's fine. That's a good guess, because they have this point here. They have sort of this pruina. I don't know if you can see here in the corner, is sort of having this like snowy pattern on the lobe tips. So that's pretty common for Pixini species. And there are some crusts, right? Some of our favorites, very difficult to identify unless you collect them. However, there are some easy ones in the area. The smoky eye boulder lichen, my very favorite. You see growing on rocks. The frosted coma lichen, the chrysotrix, is also seen in the city. And then the fluffy dust lichen, the Lepra finckii, also all of these pictures taken in the DMV area. So these were separated by, folios, crustos, fricos, but we know that they don't live like that, right, in the real world. They grow all like together. So first of all, you need to separate what's what. So in this case here, when you go up close, you see Physia. So Physiaceae is another family that's pretty common in the area. So Physia ipolia, but you can also have Physia stellaris. They're very similar, but some of these two are also some of the most common species, the Candelaria concolor, Physia milligrana. Yeah, these two are also very, very common species. And then Lecanora, even though this is Conisioides, which is supposed to be one of the most common species, Lecanora or rim lichens, they're one of the most common crustose lichens, and they all have these what we call lecanorine, excuse me, apothecia, which means that their apothecia, their border, has the same color as the thallus. So when you see a crustose lichen, like this is a foliose lichen, but you see this also has the border of the apothecia, has the same color of the thallus. That is lecanorine, and that is usually for a crustose lichen, that is usually a lecanora lichen. And some other common lichens, you see again the Candelaria concolor. Now I just wanted to show you how it can completely cover a tree. It's not just that tiny little one that you see here. See, so this is up close. So there are two main families that have these bright colors. So first of all is the Candelariaceae and the Teloschistaceae. It's not easy to at first separate them, but you get the knack of it. And Tom and Natalia and Serenella are there to help you in the field. But my first way to separate them is that Candelariaceae, for the crustose types, the usually the apothecia and the thallus is a lot, the yellow is more like a yolk yellow, while the Teloschistaceae is a little bit more yellow, but more orangish. And if you're studying lichens, you start getting a lot more complicated with the colors. You get a lot more picky, and you use a lot of ish. But bear with me and just practice a lot. Okay, so. And again, this is just a talk. It takes a lot of practice. Just go out there in the field, use the resources that you can get your hands on. Use iNaturalist, go out with people, ask people, just do your best. And then some rare lichens, but you'll still see them. You can see some graphies. So this long lirelle here, which is basically a long apothecia, excuse me, a long apothecia. You might also see something like this with small little pimples, which is mostly in the genus Pyrenula. And you might, I've seen it one time in my life there, Cococarpia, which it might still not be a Cococarpia, but it was pretty gray, blue-gray. So I'm gonna think it's a Cococarpia. So, so I hope that I was able to convey my message that lichens are composed of many components and can be studied in the context of fungal systems, photosynthesizing systems, but also microbial systems. And we do need a deeper understanding of how these systems work to advance overall basic biodiversity knowledge. And I do wanted to end by saying that I believe that science is a movement, not just a collection of facts or methods, but a dynamic, ongoing process that involves, discovery, collaboration, and the pursuit of knowledge. So I hope that if you learned something here today, you can pass on for someone else. And I hope that they can also pass on something new that they learn. So with that, I'd like to thank you all, and special thanks again to Serenella Linares, also Annie Weissman that had also reached out to me, and President Tom McCoy, and y'all for coming. Thank you so much.
Thank you, Manuela. This was a fantastic presentation. And I believe we have a couple of minutes. So if you are in the audience and would like to ask a question, please raise your hand so we can unmute you. If you're not comfortable using your voice, you are welcome to put your question in the chat and both Manuela and myself can read it. Let's see.
I got a question for you, Manuel.
Mm-hmm.
Since you're an expert on Dictyonema, I'm probably slaughtering that genus name. The only lichen that's ever been discovered to have psilocybin in it is Huarani, and I know you know a lot about that lichen. Have they ever found? Is that the only one to date that they've ever found psilocybin in? And we've never, and the only example of it we have is in a herbarium. We've never found it in a wild sense. Is that all true?
Yes. Unfortunately, we haven't kept going. We haven't done anything since then. Nobody else has done anything since then. My next step is to, okay. So sorry, I just read a message. I thought somebody had asked. So about addiction Warani, yes, all you said is true. All we know is on that paper that is potentially hallucinogenic. We couldn't compare to the real psilocybin because of, bureaucratic paperwork. We couldn't get our hands to research at that time, and we also would have to sacrifice too much of the sample. And at the time, that was all we had. I have identified what could be potentially a second specimen, but I haven't sequenced it. So there is potentially another specimen. And my next step will be to sequence. So this is part that I was saying that about the metagenomics. My ultimate goal is that because it's an old sample, so it's to sequence that, the genome of that, and then try to identify, because now they have sequenced the genomes of all of these specimens that have psilocybin. So now that they have done that research, I think it came out last year, now I can specifically look for the genes that produce that in my sample. So that is the next step, is not to try in the like wet lab anymore, is just to go for the genes to see if that they're there. So that's my next question. Thank you, Tom.
Wonderful. From Christine, we have a question. Do you ever find yourself having difficulty classifying new species of lichen? What is the most common roadblock you encounter in the field or the lab when finalizing taxonomic classification?
Yes, so the answer is yes, because there you sometimes you get overwhelmed with conflict in your data. Sometimes you don't get the results that you want, right? But you can't choose your results. You just, the result is the result. So sometimes you have a beautiful hypothesis of like, these are the same species or these are not the same species, and you don't get that. So that's why you have to be open, and you don't try to fit your hypothesis. You test your hypothesis, but you don't try to fit in what you want into your results. So that's one thing, you have to just keep your mind open. So yes, I do find conflict, and it will help me to finish responding that by the second part. You said the roadblocks. I think the roadblocks are very interesting that you asked that, because I think the most difficult part is the taxonomic mass that is out there. I think there's, the old literature, there's a lot of names sometimes available, and the descriptions that they used in the past is very ambiguous. They were not very detailed. So sometimes there are 10 names for the same thing, and the descriptions that they used could fit 10 different species, but you have to have the type. So the type specimen so when they describe each species, you have a type. So you have to request the 10 types to see which one is the representative. Am I making sense? It's just like, that's the most difficult part, because there are some herbaria that don't let you borrow types, and you have to see the types. And sometimes the type specimen is this big, and they don't have characters as well. So sometimes you just have to make a very harsh decision, which is to select a new type. Sometimes you just have to sort of, I don't know, I could just keep talking about these, but I would say that sometimes you just need to go with what you have, and and that's just how science goes. It's like, this is my data, and sometimes you just need to put it aside and say, I'll deal with it later, until you have more data, until you have a genome. But sometimes more data don't necessarily help you. Sometimes it makes things more complicated. So long story, not as responding. Yes, sometimes it's very difficult to publish a new species. So I think the best way is usually to talk to collaborators and colleagues that also, know the group, and then you can say, here's the data, here's what I found, what do you think? So I really like to have friends for that.
Thank you. Christine says that it does make sense. And then Tia asks, when working out systematic relationships between lichen groups, is there a greater weight placed on the fungi DNA, or is it truly a more even integration of DNA and all the organisms that form part of the thallus?
That is a great question, Tia. And yeah, it's usually, even though I talk about integrative systematics, you're correct in thinking that it's the fungal DNA, because we're classifying the fungal component. Because the lichen, even though the lichen is everything, when we're talking about lichen systematics, we're talking about the fungal partner, because it's the fungal lifestyle. So even though the lichen has all these different components, the name of a lichen, according to the botanical code, it belongs to its microbiome, so the mycological partner. So it's the fungal partner.
Interesting. Thank you so much for an amazing talk. That is what Tia responds. And I have a question of my own. So we have the community where is the fungus, the bacteria, and then a secondary microbiome that comes after it's been established, right? Are there cases where the bacterium can live by itself and the fungus can live by itself, or it's always lichenized?
So I will try to be short. I know I talk too much. So 99.9% of the time, the fungi depends on the lichenization to survive. There's a few examples, maybe a handful, that the lichen can survive a stage of its life not lichenized, but it's usually always has to lichenize, otherwise it will not survive. On the other hand, you mean by the bacteria, you mean the cyanobacteria or the photobiont, correct?
Right.
Yes. On that case, the answer it's not so set in stone because we don't know that much. So it's thought that they can be free living. However, there are many groups that are only known to be lichenized, and especially because everything now you have to sequence it. So, but the before the what is known is that there's not a lot of characters to identify an algae inside and outside of the talus because it's just a round green thing if it's a green algae, or if it's a bluish thing is a cyanobacteria. There's not a lot of characters. So before they could still be classified as the same thing even though they were different. But now we can sequence it. So for the things that have been sequenced, there are a few instances where the same species or haplotype has been found inside and outside. But
Very.
some are only lichenized. So both. Well, the fungi mostly lichenized or 99.9 lichenized. The photobiont could be both, but there are some clades only lichenized as well.
Thank you so much. That is truly interesting. And I have so many more questions. And I also understand that it is now 9 p.m. So I'm going to pass the baton to our president. Thank you again for
Thank you so much.
talk.
Okay. Thanks, everybody. Thanks for hanging in there. Thanks to you, Manuela. Hope to see you soon. Okay. Good evening, everybody. See you soon. Bye.
Bye-bye.